So, glycolysis. Glycolysis, two parts, two halves. Want to think of it, the first part, an investment phase. Second part, the payoff. You look above my head here. There it is. This is the series of enzymatic reactions. That's the first half, the investment. Notice, how do I know it's the investment? Well, as you see right there, ATP, ATP. ATP becomes a DP, a triphosphate to a diphosphate. So, I know a few of you are probably freaking out right now. Oh my God, how do we need to know this? Do we need to know this? No, you don't need to know this, okay? Not expecting you to keep this memorized. Now, I have a problem with this figure. Because remember I told you everything in the cell is happening in water. It's an aqueous solution. And if you take sugars, they're not the straight chain as it's drawn here. That only happens up in chemistry, so chances are this was drawn by a biochemist. But you're always going to see the sugars, if they have the length and the ability, will always form geometric shapes. Glucose. Glucose in your body, or in the cheeseburger you ate for dinner, or whatever it is you ate, will always be in this geometric shape because it's biological. Glucose will have that first phosphate group added to it. First one right there, boom. Gives you glucose 6-phosphate, okay? Even down here in biology, the naming criteria you learned in chemistry still holds true. That's the 6 carbon. That's where the phosphate's being added. So, glucose 6-phosphate. The adding of that phosphate is done by a kinase. A kinase is a type of enzyme that pops off that third phosphate from ATP, adds it to the target, to the substrate. Done. Glucose 6-phosphate becomes fructose 6-phosphate, 5 sugar. Now, the way they've drawn this as straight chains, it doesn't look like it, but you've gone from a 6 to a 6 with a phosphate group added here, to a 5 carbon. A 5-sided carbon ring with 2 carbons popping off of it. It's hard to see here when it's drawn as a 6-chain thing. And then we go and we add another phosphate. So now we have 2 carbons sticking up off the 5-sided chain. Up here is the oxygen that's normally right here. And over here, it is now, excuse me while I draw these, it's hard to draw on a pad, carbon, and then the phosphate group there, carbon, and a phosphate group there. So you now have literally mirror images right down the center. Boom. Something that can be split and give you 2 that are mirror images of each other. There's no way to sit there and cut a 6-carbon ring and get the 2 halves to be the same. So, glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-bisphosphate, and then becomes the 2 halves. Now, notice here, The two halves are not exactly the same. Okay, glyceraldehyde 3-phosphate, dihydroxyacetone phosphate. Okay, turns out, one has the phosphate on carbon 1, the other has the phosphate on carbon 3. Dihydroxyacetone phosphate, our bodies can't do anything with directly. But triose phosphate isomerase, isomerase, remember, rule of thumb, anything that ends at ase, enzyme, isomerase means it's an isomer, takes something, makes the isomer the enantiomer of it. Dihydroxyacetone phosphate is the enantiomer of glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate, as we see right here, G3P, can go through the second half, the payoff, where we get some ATP back. ATP, ATP, and look right here, NADH. Now keep in mind that one glucose became one fructose, which then got split into 2 glyceraldehyde 3-phosphate. So it's actually 2X right there also. 2 NADHs, 2 excited electrons to be used at a later point in time. 2 ATPs, 2 ATPs. What you see here, the second half, glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate, 3-glysoglycerate, 2-phosphoglycerate, phospholipyruvate, ending in pyruvate. I like to think of it as basically just rearranging the furniture. Other than the phosphates that get picked off and the initial 2 hydrogens, nothing else other than the water there is being extracted. Things are just being moved around, rearranging the furniture, so that glyceraldehyde 3-phosphate, a 3-carbon, will end as pyruvate 3-carbon, but as you look, it has a hydroxyl at one end, whereas glyceraldehyde 3-phosphate has a carbonyl group. There you go. There's the big difference. So when you look at it all together, 2 ATPs go in, 4 ATPs come out. Glucose gets turned into fructose. Fructose gets split in half, giving you 2 halves. The 2 halves get some hydrogens, electrons stripped off, water removed, and the phosphates removed. The 2 phosphates you see in the payoff phase, or the 4 that ATPs being made, substrate level, slow and clunky, even though it's an enzymatic reaction. And there you go. Glycolysis. 2 pyruvates, ready to go. You see above is a link to a YouTube video that somebody has made to illustrate the steps we've been discussing.